we talk about nucleic acids, really this, all right guys, be quiet. This really can also fit in our genetics unit, because when we talk about nucleic acids, we're really talking about DNA and RNA, because those are the organic molecules that our DNA and our RNA are made of. That's what the Na stands for in DNA, deoxyribonucleic acid, or ribonucleic acid for RNA. So we'll talk about it now. We'll also start to get into a little bit of genetics when we talk about transcription and translation. And we may come back to it as we move on and get into genetics. All right, so first off, nucleic acids are a type of organic molecule, okay, that provide for a couple important functions in living organisms. Okay. They compose our genetic code. So our DNA, our genes, the chromosomes that you inherited from your parents, the information that's stored in those chromosomes is stored in these nucleic acid molecules. And really the function, what these nucleic acids do is they specify how your cells will build the various proteins that they make. They contain the instructions for protein synthesis, how to put together. We talked about the wide variety of proteins that are produced in living things, and the instructions for doing that are stored in the DNA. So when we talk about nucleic acids, you're generally talking about DNA or RNA. They're both molecules that are similar in structure, both molecules that store information, although they have different functions when it comes to the translation of our genes into proteins. So DNA is deoxyribonucleic acid. That's the term for it. And we'll look at the structure of these molecules in a minute. And RNA is ribonucleic acid. So DNA is found inside of the nucleus of cells. There's also some in other areas of the cell. We talked about that when we talked about the heterotroph hypothesis and the evolution of eukaryotic cells, that there's DNA also in the mitochondria. There's DNA in the chloroplast as well. And so mostly though we're talking about DNA that's inside the nucleus of our cells. So if we have a cell inside the nucleus, we know there are chromosomes. In humans it's 46 chromosomes. In other species it can be other numbers. But those chromosomes, those little packages, are made of DNA along with protein. DNA contains carbon, hydrogen, oxygen, nitrogen, and phosphorus. And it's a very large polymer. Okay, so it's a large polymer, and the monomers of a DNA molecule are individual nucleotides. Okay, so the monomers of a nucleic acid are these things we call nucleotides that have 3 parts, and when they get attached, they form the structure, the shape, it's called the double helix. So if we look at nucleic acids and we look at a single nucleotide, it's composed of 3 parts basically. Okay, and so this is sort of the structure of one of those nucleotides. We start with a phosphate group, PO4. Generally, when we are drawing models of the DNA molecule, we usually draw it as a circle. So here is a circle, here is a circle. So the phosphate group, it's attached to a sugar. It's a 5-carbon sugar. So we usually draw it as a pentagram 5 carbons in the sugar. So in DNA, it's deoxyribose. And then there's what we call a nitrogenous base. And this nitrogenous base, obviously, has nitrogen as a main component of it. And just like an amino acid had a group that could vary from one amino acid to another, how many different groups were there? 20, to form 20 different amino acids. DNAs are nucleotides, have just 4 different nitrogenous bases that can be in this location, okay? And so the bases are adenine, thymine, cytosine, and guanine in DNA. There's going to be a variation when we talk about RNA.
But are they different?
They're all different.
So wait, there are only 4 kinds of like.
There's only 4 possible nucleotides in DNA. And you call them these by, they're named by those nitrogenous bases. So one nucleotide might have adenine, one might have cytosine, one might have guanine. And so the structure of this molecule is, if you think about, like these DNA molecule, which is this double helix shape, okay, a twisted ladder. Okay, the sides of the ladder, those are made of the phosphate and the sugar. And then the rungs of that ladder, these central areas, are made of the nitrogen bases. Because DNA is a double-stranded molecule made of 2 strands of nucleotides. And they match up in certain ways. They form this double helix, this twisted ladder, the spiral staircase. The structure of DNA molecule was discovered by a few scientists back in the 1950s. Very famous now. They won the Nobel Prize for their work on determining the structure of a DNA molecule. Named James Watson and Francis Crick. They also used, and a woman was vital in their work in terms of, she worked with X-ray crystallography, and Watson and Crick used some of her X-ray images to come up with their model, but really she never received proper credit for the work that she did. It was sort of shortchanged. They sort of used her work, and she did not get the recognition that they did. And we'll learn a little bit about that later on. So when a DNA molecule forms, it's double-sided. So if we go back here, what you'll notice is that there's 2 sides. So there's a sugar, phosphate, backbone. There's 2 of them. And each of them have attached to them a series of these nitrogenous bases. Okay, so it's a double-stranded molecule. Okay? And the bases always match up in certain ways. They are called complementary. So if you have adenine on one side of the ladder, the other side will have thymine. You have cytosine on one side, you have guanine on the other side. A and T always go together, G and C always match up together. Okay? And so if you look at this diagram, okay, you can see that. Here we have sort of a flattened version of a DNA molecule. 2 sides to this ladder. This would be the sugar and the phosphate. This would be sugar and phosphate. And then we have the nitrogenous bases attached. Okay? They are complementary to each other. So if we have A on the left side, we always will have T on the right side. If we have C, we have G. If we have T here, we have A here. And if we have G here, we have C there. They always match up in those ways. Okay? Now they're drawn here as shapes that match with each other, although it's more about the chemical properties of those nitrogenous bases which allow them to link up in those ways. But what this means is DNA molecules can be replicated as long as you know one side of the DNA molecule, you know what the other side should be. Okay? So if you didn't have this side, you could predict what it's going to be based on these bases, right? What would it be? T, G, yeah. So when a DNA molecule replicates, which has to happen every time the cell divides, it needs a complete copy of its genetic code. And so what happens is, okay, the DNA, we'll talk a little bit more about this later, unzips and the two strands separate. And then sort of available here are just nucleotides that are just sort of floating around. And they start to match up and bond correctly. Okay? And again, they only bond in certain ways, A with T, C with G. And once that completely fills in, now we have two DNA molecules. And they're the same. Right? We have the same bases on each side because they are complementary to each other. So that's what allows a DNA molecule to be duplicated, to be replicated. If we look at the bases, okay, the nitrogenous bases here, you could see we have thymine, cytosine, adenine, and guanine. Okay. And they come in two categories. They're pyrimidines, okay, which are cytosine and thymine. They're just made of one ring of molecules, versus the purines, which are made of two connected rings of molecules, adenine and guanine. And so what you see is that you always have, okay, one of the purines matches up with a pyrimidine. And again, what that allows is there to be consistent distance between the sugar and phosphate backbone, because we have those bases matching up in that way. They bond together with hydrogen bonds. It's what holds together the nitrogenous bases. And those hydrogen bonds can be separated by different enzymes when this DNA molecule needs to be replicated. Okay? And so it eventually forms this structure. We're not going to move on to RNA right now. We'll get to that later on. But any questions thus far about DNA?
Splitting up, like for those, yeah, when it unzips, would it keep, and then it goes back to 2, would it keep unzipping?
Yeah, it basically, the molecule becomes unzipped, separated, and as it does, the bases fill in each side, and it continues all the way down till it reaches the very end of the DNA strand, and then you're left with two identical copies of the original DNA strand. It only will replicate when necessary. So DNA replicates when the cell needs to divide, basically. Nate?
Oh yeah. So where in the ring of each of the
Yep.
so wherever there isn't nitrogen